So, Kirtan, can you confirm if the screen is visible to you?
Yes, ma'am.
Okay. So, in the previous session, I think yeah. So, in the previous session, we finished many of the topics, right? Um, can you help me remember what was the previous topic that we discussed? I suddenly forgot. Mm.
I'm not at my home
Okay,
and I'm
okay,
not. Yes, ma'am.
okay, okay. Just give me 1 second then. Right. So in the previous session, we actually finished how we will be delivering the signaling molecules, that is the growth factors, to our tissue engineered constructs. Okay. And what are the strategies that is followed to do that? So now we will discuss another topic from today onwards. And this is kind of the second last chapter of this tissue engineering course. So we will be discussing about the immune response that the body gives to the foreign body. So whatever tissue engineering construct that you will be implanting after making it, that is a foreign body, right, to our immune system. So some immune reactions takes place, and it decides whether that particular implant will be able to function well or not, or what will be the long-term effect of that implant on our body, whether we will develop some other side effects or not. So then we have to go into modulating our tissue engineering construct so that we get a favorable immune reaction. Okay, and that prolongs the life of the tissue engineering construct. So before we go into discussing the details of that, we will like briefly discuss some topics of the broad field of immunology, and we will only cover only those topics which are relevant to our further discussions. So we will look at what are the immunity types that we have, okay, then the cells, some common cells of the immune system, then what is the process of phagocytosis, okay, and also, to our discussion, the knowledge of the foreign body giant cells is important because we later on see that these are the cells which are formed in response to the large tissue engineering constructs or the scaffolds that we implant. Okay, so in response to that, these cells are formed later on. We will see how they are formed and how they act on it, and also the different kind of inflammatory responses that is seen. First, just quickly go through the terminologies. Today's lecture is going to be like that only, bit about immunology, okay? So that it will help you recall the terms, the functions of different topics like the cells and the immunity types, etc. Just a primer of immunology will be there in today's class, okay? So immunology is a field of biology where we study the immune system of all organisms. That is immunology. And we are interested in studying the immune system of humans. The term immunity refers to the protection of our body against the pathogens or the infections caused by the pathogens. So our body is very resilient against the infections, and it has a like very vast defense mechanism to fight against this. So this particular yeah, phenomena is called immunity. What are pathogens? So what are the organisms to which we are vulnerable or our immune system has to put on their guard? So usually the viruses, then the bacteria, various fungi, then parasites, and various proteins, only virulent proteins if you inject, then also some infection or disease can happen. So do these kind of pathogens, our immune system works and tries to fight them. So I have a one query. I don't know whether you'll be able to answer or not, but maybe you can think about it later. So, do you think cancer cells are pathogens?
Actually, ma'am, I think no, because a good pathogen can interchange their host, and cancer cell cannot change up to person to person. It remain in the same person which is acquired, so it is not a pathogen
So you're trying to
or
say
path.
that, yeah, so you're trying to say is like the cells that have become cancerous are the host cells. So the body um, like doesn't see them as pathogen, right? You're trying to say that, I
Yes,
think.
yes, ma'am.
Okay. So basically pathogens, we usually refer to those organisms which are external, okay, foreign. So that uh, for those organisms like these virus, fungi, bacteria, we call them pathogens. All right. Then there are various types of immunity. So one type is the innate immunity, another is the adaptive immunity. So innate immunity is our body's primary form of defense mechanism. Or you can say it is the immunity first which acts against the pathogen. Later on, the adaptive immunity comes. And it is present from birth. So from our childhood, the innate immunity is there from the day we born. It is responsible for giving an immediate response. So whichever pathogen attacks, so the components of the innate immunity first goes and tries to fight it. So what are the components of the innate immunity? First is the barriers. So our body has like the organs of our body, they have barriers, right? So first barrier that we all know is the skin, which is the outermost layer of our body. So it is a barrier. It protects the inner organs, the visceral organs from any damage, right? Then there are mucosal membranes, like in various canals in our body, like nose, then gastrointestinal tract, then eyes, lips. So in our mouth as well. So the mucosal membranes, they produce mucus, and this mucus, it can trap the pathogens, even not only pathogens, some dirt, etc. So hairs are also there along with mucus. So they function as the barrier. Okay. Then the pH of our stomach. So somehow, if we ingest some pathogen through our food or drink it in our like water, etc. So the pH, which is around 1 to 2, which is highly acidic, uh, that pH is detrimental. It can kill those pathogens and won't allow them to attack any of the organs. Then the microbial flora in our gut. So our gut has various types of microbes. Now some of them are good microbes, and these good microbes secrete various kind of proteins, and these proteins participate in various biology pathways, biochemistry pathways. Okay, so through these good bacteria, the proteins that they secrete, they can basically hinder the activity of these pathogens if at all they enter the intestine after going through stomach. And apart from the barriers, if some pathogen enters in your bloodstream, say you prick some accidentally, you like got pricked by a needle, okay, or some cut from a rusted metal, something like that. So those surfaces contains large amount of bacteria. So and they can easily enter our bloodstream. So if so that is why the bloodstream also has various kind of cells floating in it that can actually act on these pathogens who enters in our bloodstream uninvited. Okay. And apart from the blood cells, there are also complement factors that are present in our blood serum. Those also help us to fight against those pathogens. So, so these are the three components of innate immunity. Apart from that, this immunity is very non-specific. So it doesn't see or doesn't recognizes the pathogen. It just see that something foreign, anything, a cell or dust or protein or any material has entered your body. So it will perceive it as a pathogen and start acting on it. So it will either try to engulf it, release some enzyme, hydrolytic enzymes to destroy it, okay, and then degrade it. So that is its response. That is the way the innate immunity works. So it is highly non-specific. Just remember the term non-specific. Whereas adaptive immunity, it is very specific in the way they act, okay? Means they first work to the components of the adaptive immunity will try to first recognize, and depending on what it is, it will engineer a appropriate immune response, which is why the response is delayed. Okay, so the major cell type of adaptive immunity is the lymphocytes. And we know there are three types of lymphocytes: the T lymphocytes, B lymphocytes, and the natural killer cells. So these three types of lymphocytes, they are responsible for the delayed response, and together they are the component of the adaptive immunity. B cells are responsible for producing a protein called antibodies, and these antibodies, are produced very specifically to bind to a specific antigen. Okay, by the T cells has various types of subtypes. So there is not one type of T cells, there are different kinds, like the effector T cells, helper T cells, cytotoxic T cells. Okay, and there is another type of cell that is called natural killer cells. But these types of lymphocytes are a bit different from T and B cells. How? We will later come to know. And both innate and adaptive immunity, they can give cell mediated response, because innate immunity, you saw there are various kind of blood cells, right? As well as adaptive immunity has various kind of lymphocytes which act and try to kill the pathogen. Whereas humoral immunity or humoral based immune response, we refer to the immune response which is mediated by the antibodies, not cells. So immune response here we can again distribute like cell mediated and the humoral is antibody mediated. Okay, so these are the three line of defense. Yes, any query?
No, ma'am.
Okay, so these are the three lines of defense system that our immune system has, means three lines of defense. First is the barriers, the physiological barriers, that is that the pathogen has to pass in order to enter our body, like the skin, then the mucosal membranes, as I described. Then in the intestine there are cilia. Okay, so they are like hair-like projections that are there, completely covering the inner lining of small intestine. So they can also trap the pathogen, okay, and stop it from going forward. Then the temperature of our body. So it can basically kill those pathogens which cannot survive at this temperature. Then the pH. So the acidic pH of our stomach. So these are all physiological barriers that our body has. If suppose any pathogen passes through these physiological barriers and enters our body, then it has to face the second line of defense, which is the innate immunity. So in innate immunity, as I said, there are various kind of blood cells. Okay, so the neutrophils, basophils, eosinophils, macrophages, mast cells, dendritic cells, natural killer cells. So these are all the cells which are present in the blood, as well as few of the cell populations are also present in the tissue. And suppose the pathogen is still resilient and still fights these blood cells, and still like living, a few of them are still surviving, then it has to face the final line of defense, which is the adaptive immunity components, where there are the B cells who produce various kind of antibodies, and there are different type of T cells like cytotoxic T cells, helper T cells. So they then engineer a very hard line of defense, which that pathogen can hardly pass. Okay, so at this point, most of the pathogen will get killed. So these are the three lines of defenses that our immune system has. Now, most of the cells, or most of the cells of innate immunity, they exercise a mechanism called phagocytosis. And what is phagocytosis? It is a process by which these cells, or the phagocytic cells, can engulf. They basically extend their membrane in form of lamellipodias, okay, pseudolamellipodias, and encase the pathogen around them, and bring it into its cytosol. Okay, so in the cytosol, there will be various kind of hydrolytic enzymes which will kill it and degrade it. So that is phagocytosis. So what are the phagocytic cells? So the macrophages, neutrophils, monocytes, and the tissue macrophages. So these all cells are phagocytic in nature. And what are the four main attributes of adaptive immunity? So how will you categorize them, or how will you characterize, not categorize, how will you characterize the adaptive immunity? First, this immune system, or the components of the immune system, they will only act depending on what kind of antigen it is present. Okay, so they will recognize the even the minute differences in the antigen, and for each type of antigen, it will try to secrete different antibodies. Okay, so they are very specific in nature. In that way, the specificity come. Then diversity. So for different kind of antibodies, so different kind of antigens, there will be like all the different unique types of antibodies. For each type of antigen, there will be one antibody. So uh, this immune system, that is adaptive immune system, it has means large collection of antibodies present, which can like recognize any of the antigen that is coming in our body. So it gives us a like very strong defense mechanism, if you if that makes sense to you. Okay, then this immune system also has immunologic memory. So if suppose you're attacked by some pathogen few years back, so the components of adaptive immunity, they are means the cells of this adaptive immunity, few of the cells, they are called memory cells. So they will remember that attack. Okay, the next time you're attacked by that same pathogen, these memory cells will trigger the B cells to produce the same kind of antibody, and do it in a much larger quantity, means maybe 2x times antibody production will be there. So the next time when pathogen came, it was killed or it was neutralized much faster. So this through this immunologic membrane, the effectivity or effectiveness of our immune system has become much more high. And then uh, innate immunity cannot really recognize that well between self and non-self, but self and non-self recognition and the effect thereon is better by the adaptive immunity. So these are the four characteristics of adaptive immunity. Now we will discuss what are the cells so of immune system. So the cellular components, and we will touch upon deeply onto those cell types which are relevant for our later on discussion on host response to biomaterials or tissue engineering constructs. So this is the bone marrow, and it is the site of a very important process called hematopoiesis. So you already know this. So hematopoiesis is a process where blood cells are formed. So at bone marrow, there is a stem cell called hematopoietic stem cells. Okay, hematopoietic stem cell. So this stem cell, it can give rise to two types of progenitors. So it can divide and differentiate to give rise to two types of progenitors. So one is the myeloid progenitor cell, another is the lymphoid progenitor cell. So from our discussion on stem cells, I hope you know what are progenitor cells. Now, the myeloid progenitor cells, they can further divide and differentiate to give rise to various type of mature blood cells like the RBCs or the erythrocytes, then the eosinophil, basophil, neutrophils, then monocytes, megakaryocyte. So these megakaryocyte, it further differentiates to give rise to platelets in which is found in our blood. Whereas the monocyte, it can differentiate to give rise to either dendritic cells or the macrophage. And the lymphoid progenitor cell, which was developed from the hematopoietic stem cell, it can give rise to another set of cells, which are the T cells, B cells, and the natural killer cells. So the B cells, they actually then become larger and become plasma cells, and these plasma cells through are like very high, or they will secrete large amount of antibodies. Okay, so this is the hematopoiesis process. So now you know. So the same thing is written here. What are the means from the progenitor cells? What are the differentiated cell types that are developed? Okay. Now, the natural killer cell, as I mentioned in my previous discussion, is different from the B cell and T cell. So it is also a lymphocyte because it has developed from the lymphoid progenitor cell. So natural killer cell, but it is different from the T cell and B cell. How? These natural killer cells, they do not have the cell surface receptors through which it can recognize antigens, whereas T cell and B cells have these surface receptors. So in this way, these are different. And what are the major organ systems in our like major organ systems which are responsible for the immune system, or where the immune cells develop? So, in the bone marrow, the hematopoiesis process happens. So here the blood cells means these blood cells, and uh, these T cells, B cells, they are formed. But further maturation of T cell, it happens at thymus, but B cell matures at bone marrow. So bone marrow and thymus are the primary organs where these immune cells are formed and subsequently become in its mature state. And there are secondary organs like the lymph nodes and the spleen. So here you can say where, at the locations where say macrophages recognizes the or engulfs means phagocytosis is the pathogen. The further degradation where it is usually happening. So it usually happening at the lymph nodes. And lymph nodes are connected to our body's lymphatic system. And lymphatic system is nothing but the sewage system of our body where all the waste gets like collected and subsequently removed. So that is why the lymph nodes are like the final death point or the grave for these pathogens. The same is true for the spleen. Okay. Now, what are the if I take 100 white blood cells? So let me just give you another classification of these cells. So cells which are not red, like the T cells, B cells, then monocytes, neutrophils, basophils, eosinophils. So these are not red. So in general, people call them as white blood cells. All right, and the red blood cell is simply erythrocyte. So in this way also you can classify these immune cells, and also depending on what is the lineage you can also classify these cells. So if I take 100 white blood cells and see the percentage of different kind of white blood cells, then what will be the like finding? So we find that neutrophils comprises around 60 to 70%. Okay, and next highest number is of the lymphocytes, means the T cells, B cells, natural killer cells together, and the remaining is composed by the basophils, eosinophils, okay, and monocytes. So these are the remaining white blood cells types. And monocytes, they can basically further differentiate to macrophages. That is why macrophage is not mentioned here. So among these cells, what are the phagocytic cells that I previously discussed? So these are the neutrophils, monocytes, then eosinophils. So I should have written monocytes as well. So these three cell types are the phagocytic cells, means they can do the process of phagocytosis. Whereas basophils, it is not phagocytosic. Also lymphocytes, they cannot phagocytose. So this is how the percentages look like. And we can give another classification basically, depending on how these cells look like when appropriate stains are given. And um, when you after staining, when you look under microscope, they give some characteristic features. So based on that, they people have like given classification. So white blood cells is also called leukocytes. And we can again distribute these white blood cells into either granular cells or non-granular cells. So what are granular cells? So those cells which under microscope looks like there are small packets of something floating around in its cytoplasm. So granules, people call them as granules. So cells having granules, so that is called granular cells. Whereas cells which look under microscope relatively plain, there is nothing floating cytoplasm, those are called non-granular cells. And uh, granular cells can be further classified into three types, which is the eosinophil, neutrophil, and the basophils. Now, how these three are different, I will come to it in the next slide. Whereas the non-granular cells, the lymphocyte and the monocyte. So, like depending on whether the cells are phagocytic or not, we are classifying. Then the type of staining characteristic the cell has, we are classifying. So also the color, we see the erythrocytes, but the white blood cells. So depending on the color as well, they are characterized. So the lymphocyte, the T cell, B cell, as well as the natural killer cell that is not mentioned in this figure. And the monocytes, it can differentiate to become either macrophage or the dendritic cells. So I was now going to tell you how the granular cells are classified. So the granular cells, the basophils. So why we are calling basophils? Because these cells can be stained with only a basic dye, which is called methylene blue. That is why people have given the name basophils. Whereas this neutrophils, these cells under microscope, when observed, they are they contain various kind of nuclei, means various large number of nuclei. So here, this is the neutrophil. So this red, like purplish, is the nuclei, and you can see the nuclei is like divided into three compartments. These are called lobes, which is why neutrophil has a multi-lobed nuclei, and it stains with either basic and acidic dye. So neutral. It is not just responding to one type of dye, but it can respond to both acidic and basic dyes. And eosinophil, it has bilobed nucleus. So in this figure, you can see there are two lobes in in its nucleus. That is why it is bilobed. And since it stains with only acidic dye, and the name of that dye is eosinophil red, which is why its name is eosinophil. So you can see how these three cell types of granular characteristics are named, and depending on their staining characteristics, means how they stains. Then mast cells, they are also granulocytes, and they are found. So in this figure, it is not there, but just for your information, mast cells, which is part of the innate immunity, they are also granulocytes, and they are generally found in connective tissues like the dermis layer of the skin, or etc. Like all the connective tissues like bone, muscle, tendon, etc. And their appearance is quite similar to basophils. So this is similar to this, the neutrophil mast cells look like. So I think in one of the figure there was mast cells. So yeah, so this is mast cell, and this is basophil. This is how, like, the only difference I think is the basophil has bilobed nuclei, and mast cells, they do not have bilobed, but it is granular. So you can say these are granular, the dot dot. No, this is macrophage. Oh, this is mast cell. Sorry, this is mast cell. These are like have a granular characteristics. Neutrophil, then basophil, eosinophil, and mast cell. All right. So this lecture, if you are feeling like bored, it's totally fine. Okay, because I think many of you have already studied immunology in your courses, respective biotech courses. But I needed to give you a primer, because if I don't give, and if you don't do your own homework to go through the basics on your own, then you won't be able to understand the next topic. Okay, that is how our immune system basically acts on something foreign, that is a biomaterial scaffold or a tissue engineering scaffold, how that interaction takes place between biomaterial and our host and our tissues. So for that understanding, I'm giving you this primer. So now we can like classify these types of these immune cells into whether they are in the adaptive immunity or whether they are in the innate immunity, and you will find there are few cells which lies in the overlap. So the natural killer cells, which is a lymphocyte we discussed, is like belongs to both innate immunity as well as the adaptive immunity part. And there is another type of T cell, gamma delta T cell. So this type of T cell, which is a lymphocyte again, it also belongs to innate immunity and adaptive immunity. Now, you are also seeing some protein called complement protein, that is part of innate immunity, and the proteins like antibodies, that is part of adaptive immunity. And all the other cells, I think we have already covered that they are part of adapt innate immunity. So B cell, T cell, just they belong to adaptive immunity. Whereas if for innate immunity, the macrophages, neutrophils, then monocytes, okay, dendritic cells, so which has phagocytic characteristics, they belong to innate immunity. Just remember these points. Okay. Now, what are macrophages? Okay. So just briefly, so macrophages are nothing but enlarged monocytes. So there are various type of, so there are monocytes, as soon as they are formed in the and matures in the bone marrow, it comes into circulation and it moves around for around 8 hours before many of the monocytes, they fuse together. Okay, like 3 4 monocytes, they fuse together and become a large macrophage. Okay, this macrophage is around 5 to 10 folds larger than a single monocyte. So not 2 3 monocytes, minimum 5 monocytes they have to fuse in order to become one macrophage. Also, the macrophage, the final macrophage that is formed, the number of organelles is increased in comparison to the monocyte, and the complexity means cellular architecture, the complexity in that it's also increased. The number of lysozymes means lysosomes, that is the organelle present in the cytoplasm containing large amount of hydrolytic enzymes. So these lysosomes number is also increased in macrophages. Neutrophils, they do not really produce hydrolytic enzymes, but the way the neutrophils phagocytose means engulf the pathogen is similar to macrophage. So in this way, neutrophils are different from macrophages, but both are capable of doing the phagocytosis process. Now, there are few macrophages which are found in our blood. Okay, so they are floating in our blood, but there are also those macrophages which are tissue specific. Okay, which are tissue specific, like the alveolar macrophages, which is there in the lung. Then Kupffer cells, I think you might have heard. So this Kupffer cells are present in the liver. Osteoblast, they are the macrophages found in the bone. Microglia is present in the central nervous system, okay, basically in the brain. Then not in spinal cord, but in the brain. Then histiocytes, these are present in various connective tissues, like maybe not bone, maybe cartilage, then tendon, okay. Then there is mesentelia cells, so these are found in the kidney. Also, um, there are there are few cells, few tissue specific macrophages found in the skin called Langerhans cells. So this is a diagram showing this, say, this particular macrophage which is there in the blood. It goes and binds, enters a specific organ or a tissue, and decides to become a resident of that particular tissue, and this become tissue specific macrophage or tissue associated macrophage. So in the brain there is microglial cell, as I mentioned. In the lung there is alveolar macrophage. In the spleen also there are macrophages like spleen red pulp macrophage, marginal zone macrophage. Then the peritoneal cavity, so there also there are few macrophages like the peritoneal cavity macrophage, and the skin there is Langerhans cells. So these, so our innate immunity is present everywhere. You can say it is present at the organ level, as well as it is present in the bloodstream. So how macrophages are maturing? Now we will discuss that. So in this figure, you see this is the myeloid progenitor cells that have formed from the hematopoietic stem cells. Now these myeloid progenitor cells, they can like say one of the daughter cells of that myeloid progenitor cell, it has decided to go towards differentiation of a monocyte. So it can become a monocyte or a monocyte. So this monocyte, it will then like fuse to become macrophage. That is how it goes. So I have seen the term monoblast in various literature to refer to the daughter cells of these myeloid progenitors who have decided to go towards the formation of monocyte and subsequent macrophage. So from myeloid progenitor cell, the monoblast is formed, and then it forms monocyte and then macrophage. Also, the macrophage, it can either go and become a tissue specific macrophage, or so this is a tissue associated macrophage, tissue specific macrophage, or there can be macrophages who are like like perpetually becomes an activated macrophage. So there are two types of activated macrophage that is known. It is M1 macrophage and M2 macrophage. And these tissue specific macrophages, you can call them as resting macrophage. So they don't really act until and unless some signal is received. Whereas activated macrophages, they are always on the lookout. They are the sentinels, like the guards, and they are floating in our blood. So if any pathogen come, they are the first to arrive and kill them. So these are the different kind of macrophages that are formed from the maturation step just I explained. So the resting macrophages, if they receive an stimulus, like stimulus of what? Stimulus that a pathogen has entered your territory, say, then only it will get activated and act against the pathogen. So uh, the macrophage, the stimulus for the macrophage, for the resting macrophage, is phagocytosis. So the neutrophils, they are in the blood. So they can phagocytose very nicely, but they cannot degrade. So what they can do, they will phagocytose that pathogen, uh, and because, and then they will signal neutrophils, they usually start secreting certain cytokines. So these cytokines can then reach these resting macrophages, right, and uh, activate them. So phagocytosis in this way is acting as an important stimulus for these resting macrophages, which is there in our tissue. Then the cytokines, which are present, which are secreted by the T helper cells. So T helper cell is a subtype of the T cell. So they can also signal these tissue-specific macrophages to become active. So the innate immunity and adaptive immunity, they don't act independently. They act in a coordinated fashion. The components of innate immunity take help from the adaptive immunity and vice versa. The activated macrophages, they're active throughout. Okay, so they don't need any lag time to become active, which is why they are more effective. The phagocytic activity displayed by these macrophages are much more. Okay, they will secrete a large amount of inflammatory mediators. These are nothing but signaling proteins. Okay, and the cytotoxic proteins, they will also produce in a large amount. So these cytotoxic proteins are basically those proteins that are targeted to kill certain bacteria, virus infected cells, or even cancer infected cells. Okay, so there are certain type of cancers which can come from viruses like retroviruses. So in that case, if our immune system initially will try to fight it, so in that cases, these activated macrophages will try to fight. Then phagocytosis. What is phagocytosis? So I've like uttered this term so many times till now, just to tell you that it is a process that is engulfing. But how the engulfment happen? By these macrophages. Now we will come to know that. So phagocytosis is a type of endocytosis. So endo and exo, there are two ways the cells take up or release its components. Endocytosis means the cell will take that particular particle inside it. Whereas for exocytosis, the cell is trying to give out some vesicles or something like that outside in its environment. But phagocytosis is like here, what the cell is doing, it is basically engulfing entire entire cells, like microorganisms, then the dead cells. It can do that. Okay, so the whereas in other form of endocytosis, it can maybe engulf of particles up to some smaller sizes. So in this way, phagocytosis is bit different. And the in endocytosis, something called endovascular sorry, endosome is formed, not endovascular retinome, sorry, endosome is formed. Okay, and now here, whatever engulfed particle and the covering outer covering around the engulfed particle, so that entire structure is now called a phagosome. So that is the difference between normal endocytosis and the phagocytosis. Phagocytosis here, entire organisms, entire cells basically, they are engulfed. So there are three steps to endocytosis. First, the pathogen has to recognize, means sorry, first the phagocytic cells has to recognize that it is a pathogen. First recognition step should be there. But note, it is not specific. Recognition doesn't happen in a specific manner. It just sees the phagocyte means the phagocytic cell cannot really use the receptors on it to identify. So here the role of adaptive immunity component, there they will help them. Okay, so help them to recognize that it is a foreign thing, it is a pathogen. So here recognition step will happen, but the in macrophages or the phagocytic cells, they will take help from the components of the adaptive immunity. Okay, then the neutrophils attach and they will engulf. The first cell type which will do the phagocytosis is the neutrophils. But remember, neutrophils that do not have those hydrolytic enzymes to degrade it. So for the degradation, will then be done by the macrophages. So the macrophages will come here and will try to kill it. Okay, using their hydrolytic enzymes, which is secreted in the lysosomes, and ultimately neutralize that pathogen. So these are the three steps: recognition, neutrophil attachment and engulfment, and then killing and degradation. Now note the biomaterials, or the biomaterial scaffolds that you will be like making and subsequently implanting on an injured area. So those are foreign, right? So our body recognizes that as foreign. But these are very big for these phagocytic cells to engulf and further degrade. So which is why the phagocytes, they become frustrated and give a very intense phagocytosis response, and that is called frustrated phagocytosis. So that we will come to later when we'll discuss the next topic, which is the host response to the biomaterials. Okay. So in this diagram, it is showing the steps which I just discussed of phagocytosis. The first step, what is the first step? The pathogen is coming and it is getting attached to the phagocyte, means the a neutrophil. It will start with the neutrophil. Phagocyte is a neutrophil here. Then it will be ingested, means phagocytosis, engulfment is happening. Engulf here the membrane, cellular membrane, it will try to make a covering around it, and subsequently it enters cytosol, and this structure now will be called phagosome. This phagosome is containing the pathogen. Now it will be then further neutralized. So this neutralization will be possible by those cell types which is having the lysosome. So it will happen then in macrophages. So there will be like neutrophils as well as the macrophages as well. Both, they will come and like act on these pathogens. Okay. Now these enzymes which is coming from these lysosomes, they will degrade it. So degraded the partially so the components of the pathogen which cannot be digested further, they will be now eliminated as waste. So where this waste will go? This waste will go in the lymphatic system, which is why this process, this neutralization process, or the phagocytes will do this neutralization process in the lymph nodes, that is secondary organs. So in the lymph nodes, if it happens, this waste material will enter the lymphatic circulatory system and will be eliminated out from our body. Then, as promised, I was going to discuss what are foreign body giant cells. So what are these? So these are very large cells, means they are larger than a single macrophage because they are formed by fusion of large number of macrophages, and they are only formed when a biomaterial is present. So just now I mentioned no that biomaterials are larger in size than a single virus or a bacteria. So the macrophage, a single macrophage, they get frustrated. They try to engulf, but they cannot. So what they do, they will start fusing and create a giant cell that is called foreign body giant cell here. And in an attempt to engulf it. Okay, so in normal immune response, you won't see these foreign body giant cells forming. These are only formed when large biomaterial constructs like the tissue engineering scaffolds, etc., are transplanted. And what happens after formation of foreign body giant cells? Again, we will discuss it later. So as I already mentioned that they cannot be, these biomaterials, they cannot be engulfed by phagocytosis by these foreign body giant cells. So these giant cells, they basically stay nearby, like in the surrounding of that biomaterial, and slowly they try to act on it. So our body, what they do, they like see this thing is going on for so long, and then they and then the like neighborhood cells, they will start secreting some proteins and create a capsule. So this phenomena is called fibrosis. So a fibrotic membrane kind of thing is created, and it is made isolated. Okay, so that its effect cannot be felt in the rest of the body parts. So we will see that. Okay, and some and the reaction that is continuously going on near the biomaterial, that is called foreign body reaction, because it is mediated by these cells, foreign body giant cells. Now, inflammatory responses. So, um, we will now discuss the steps. What are the main steps that will help you understand that inflammatory response is happening, and also the types of inflammatory response. We will discuss that. The inflammatory response is usually localized and non-specific to a non-specific response to an infection or a wound. Okay, so it is limited to a local region. It is not a systemic thing. It is not happening throughout the body, but inflammation happens at a localized region, and it basically is a non-specific response. So anything foreign, doesn't matter what, like how, if two foreign bodies are there, means foreign materials are there in our body, both foreign materials will be given the same inflammatory response. Our body cannot basically give unique inflammatory responses to different type of foreign bodies. If you understand, that is the non-specific thing that is I'm trying to mention. The tissue damage that is happened, or that has happened due to some pathogen, okay, that has caused an infection or a wound, okay, some severe damage, tissue damage has happened due to an accident. So this tissue damage, it will start this inflammatory response. Okay, it provides this inflammatory response. Why it happens? Okay, why it is important, why our immune system like gives this inflammatory response? It gives it because it tries to protect the like it tries to like protect our healthy tissues from that damage. It wants, our body wants that that particular damaged area, it shouldn't spread. All right, it tries to restrict that damage to that particular site only. It doesn't want that other parts of our body gets also affected, which is why inflammatory response is important. The tissue damage and the infection induce some leakage of blood fluid, that is vascular fluids, that will contain some serum proteins. So in the serum proteins, some complement factors, coagulating factors, etc., those are present. And as well as there are various proteins with some antibacterial activity, like instantly if some bacteria is has come, some antibacterial proteins, they will try to neutralize it. Okay, and along with the leakage of the vascular fluid, there will be influx of phagocytic cells, that is the macrophages, neutrophils, monocytes, etc. They will try and come at the affected area, so they are the first cells to come. That is why, and their response is non-specific. So inflammatory response basically start localized region, and it is non-specific. All right. Now, there are four hallmarks of inflammatory response. How will you know inflammation is going on at your cut site? So, you will know from the color of that site. If it is red, then uh, inflammation is still going on. If you see some swelling, that is called tumor. So some Latin words are given to these hallmarks. Redness is rubor, swelling is tumor. Then often we see some that area where the injury has happened, it has become bit warmer than the rest of our body. So that is heat, calor is there. So much more prevalent is when you get a mosquito bite and that area swells up. It often becomes red if you itch it more. And if you try to feel it and focus on how warm it feels, it will be warmer. That is the like simplest way of explaining that inflammation is going on at that particular area. And yes, you will feel some pain. So it hurts more when an ant bites, a red ant bites. So then you will be relate that that particular area inflammation is going on. So these are the four signs of inflammation. So the four signs that I just discussed, why they are happening, or what is happening at that particular area, which is resulting in those symptoms of inflammation. First is uh, like the cells that has come there at that particular area, they will secrete various kind of signals, chemical signals. So these signals, they are called inflammatory mediators. All right. So what are these chemicals? They are named as histamines, prostaglandins, and kinins. So these are the three main types of inflammatory mediators, which are released by the cells at that injured site. And we can actually test the presence of these chemical mediators to confirm that inflammation is going on. So it is important when people go to a doctor and say that their wound is not healing for like years. So it is happen. So chronic. So people then try to doctor then will try to give some test to see what take a sample of that area and see what all chemicals are there. So they can say that then they finally say that this wound has become chronic now, it is not going to heal on its own. Inflammation is persistent. So then people has to assign some drugs or some other type of surgery or treatment, etc. Then at the site of inflammation, vasodilation also happens. So what is vasodilation? The term vasodilation means dilation of blood vessels. So when the blood vessels dilate, means its diameter becomes bigger, then the flow rate of blood increases. So increased flow of blood is there at that site. So this increased flow of blood makes that area red. All right. It gives a slightly reddish appearance, redder than the surrounding area. Then due to high flow, temperature, local temperature also increases at that particular area. So this increased temperature makes that area
Ma'am, you are not audible.
Is ma'am there?
I think she leave.
Ma'am is trying to reconnect. Wait.
Hello, I think I have joined now properly. I have connected using my mobile internet. Wi-Fi seems to be like unstable now. Am I audible now?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Hello, am I audible?
Yes, ma'am, you are audible.
Okay, yeah, okay. So let me again share my screen. So what was the last thing you guys heard? Were you able to understand all these three points? Like this was the last slide, no? From which I
Yes,
disconnect
ma'am.
after
Vasodilation. I
yeah.
think
So
vasodilation.
okay. So I think up to edema I discussed. So from here you guys didn't hear anything because I disconnected, correct? Can anyone confirm that did they hear anything from this slide? Did they catch any of the information that I was going to give from this slide?
No, ma'am.
Okay, all right. So I don't know why this internet connection giving the problem today. That too. I don't know why. So now I am like back. So as I was discussing, these are the signs which or these are the reasons which results into those hallmarks, that is redness, swelling, heat, and pain. So influx of another reason why we see those hallmarks is the influx of phagocytes. So due to stretching of the blood vessels, uh, cells, the phagocytic cells, they can come out and reach the site of injury. So how do they do that? So this figure will help you understand. So this is the blood vessel and this is the site of injury, and some bacteria has entered and like crossed that skin barrier. Now what has happened? These are the yellow colored cells are the phagocytes. So they will first adhere to the endothelial wall of the blood vessel through a process called margination. As soon as they do that, the due to stretching of the blood vessels, some gap between the cells get created. So these through these gaps, the macrophages, the phagocytic cells, they can come out, and this process is called extravasation. So when we were discussing about direct cell-cell contact and the malfunctions therein, we discussed the phenomena of extravasation, if you remember. So here, extravasation is happening, and the phagocyte, it will now try to uh, like sense the chemical signals, right? And according to its gradient, it will migrate at the site of injury. Apart from the phagocytes, due to permeability, so these gaps in between the endothelial cells, it is increasing the permeability of other proteins like complement antibody proteins, C-reactive proteins. So these are all proteins which are present in the serum of the blood. So they will also go and reach at the site of injury. So in this way, uh, the main components of innate and adaptive immunity, they will come together at the site of injury and do their respective functions. So margination is happening of the phagocytes. Then extravasation means it will try to migrate in between the endothelial cells. It's just like you're present in a crowd and you want to go at the first, so in Durga Puja and all, so there are so many people in the pandal, all right, and you have arrived very late. and you see so many people in front of you and you want to go in the front, so what you will do, you will try to push through uh, 2 3 people and move forward, right? So that is extravation. That is what these phagocytes also do. Then there will be migration, that is chemotaxis. Chemotaxis is basically cell migration in presence of a concentration gradient of the chemical signals. And these chemical signals which are already like present, they will give a gradient to these phagocytes. Now, as phagocytes accumulate at the site of action, they will phagocytose the bacteria, whichever has like entered that injured site, and secrete their hydrolytic enzymes and kill it. The accumulation of dead cells, the digested material or partial digested material, and the fluid, all these accumulation, that is the substance that we see pus, right? So in pimples, etc., you see white colored membrane like structure forming. When you rupture it, some white color substance come out. So what it is composed of? It is composed of these things: that dead cells, digested material, and additional fluids like serum fluids, etc. So they will create the pus. Now, inflammatory mediators, let me go slight detail into the roles of each of the types of inflammatory mediators. So histamine. So histamine basically helps in increasing the permeability of capillaries. These protein molecules, they will go and bind to the cell receptors, cell surface receptors of the endothelial cells, right? That is there in the capillaries, and cause them to dilate. Okay, signal them that it is now time for you uh, to dilate. So the diameter of the blood vessel, it increases, and thereby the it allows then passage of the serum fluids, etc. Then the kinins. So these proteins, they are present in blood uh, in inactive state. So let me just correct what I said earlier. These inflammatory mediators, they are found in blood, like in the protein form. Okay. Now the kinins, so they are present in inactive form in the blood, and they will also help in vasodilation. Whereas bradykinin may be a type of kinin. These are the proteins which activates the pain receptors in the skin and gives you that feeling of pain when like something, when you get like injured at some location in your body. That pain feeling is because of this particular protein. And that pain is important because it tells us basically to protect that area well. You should know that area is vulnerable. Now it is time for you to protect it, so that it doesn't, because that area is one like it doesn't get any more worse. So these proteins help give you that pain feeling. Another type of protein, like the C-reactive protein, serum proteins, basically acute phase proteins. Collectively, they are called acute phase proteins. So these proteins, they come out as exudate, as I mentioned in the previous slide, and reach the site of injury to activate the complement proteins. So these complement proteins, they also help in fighting against the um, like pathogen that has entered. And these complement proteins in turn are activated by the C-reactive proteins, etc. In addition, the process of inflammation also helps enzymes that are involved in blood coagulation cascade to enter the tissue. So, why coagulation is important now? It is important because the blood vessels has ruptured, right? Means so the blood can move everywhere and make the tissue area bloody, right? It will cause unnecessary loss of that blood. So we have to stop it, we have to coagulate it, we have to stop its flow, which is why the coagulating cascades or the clotting cascades, they have to be started. And how they will be started? It will be started when the components of the inflammatory process is acting on it. Means they are going to give some signals to the components of the clotting cascades to start the blood coagulation process. So once the inflammation process is over, how you will know it is over? When the macrophages and the various kind of phagocytes and the complement protein, their concentration starts declining or their activity starts declining because they have now neutralized that danger. Okay, so which is why inflammation has stopped. Then only the real tissue regeneration process will begin. So at that tissue area, the damage is still there, no, means although the pathogen is removed at the damaged area, but that tissue gap or whatever has to be filled. So how will it will be done? So the host, the resident cells of that particular tissues, they will proliferate. Okay, they will proliferate and secrete the ECM proteins. Okay, and try to cover that area, and further remodeling, tissue remodeling will happen, and together it will try to reconstruct that structure, but it will not be as like it will not be exactly the same the way it was before the injury, but it will be something similar. Okay, so that is the tissue regeneration process that will happen after inflammation is over. Now inflammation can again be characterized on like two types based on the uh, extent of inflammation. So acute inflammation, it is basically happens for a short period of time. Okay, so it lasts for few weeks, and within few weeks, if the immune system is able to clear out that pathogen, then inflammation step is over. But sometimes our body takes longer time. So when it that happens, that is called chronic inflammation. So it can take like more than a week. It can take months or years. So chronic wound gets formed. So inflammation is persistent. Okay, it is going on for a very long period of time. So what are the cell types that are involved in acute inflammation? So uh, neutrophils are the primary cells which whose concentration is very high in the acute inflammation, whereas in chronic inflammation, the other macrophages, which are sorry, other phagocytes, which are macrophages, monocytes, lymphocytes. So in under lymphocytes, there is natural killer T cells. So it is referring to those like macrophage, as it is referring to those type of phagocytes, as well as those lymphocytes like T cells, B cells, who can secrete antibodies, as well as they have T cell receptors, etc. So the entire class of lymphocytes, the monocytes, and the macrophages, these three cell types, they are basically working in this chronic inflammatory phase. They are needed, because their function is superior than neutrophils, right? So neutrophils in some ways like not that effective as a macrophage, and since this infection due to whatever reason is not getting healed quickly, which is why you need like the bosses now to take care of the issue. So these are the two types of inflammatory responses, acute and the in chronic inflammation. So that's it for today's class. In the next class, I will start the second topic of related to this only, where we will discuss how these particular components, like the macrophages, then the inflammation, how the inflammation proceeds, or how the immune system, innate immunity, etc., how they work when a foreign body. Now here foreign body is the biomaterials that is we are going to make the scaffold out of. Okay, the scaffold containing cells, that is foreign to our body. Now how our body is going to react to it, and whether we can somehow modulate it to our advantage. So that thing we will discuss in the next class. So if you have any queries, so let me know. Any queries regarding any of the topic? I think our class, like today's class, may be a revision of immunology for some of you, and I thought it is necessary because it will help you. It will give you a better understanding of whatever topics that we will discuss later. Any query so far?
No, ma'am.
Okay. Others? No query? All right.
No, ma'am.
Okay. So let me then so many of you joined late in today's class. I don't mind, it's totally fine. Just a request to join in time so that you don't miss out, right? Means you are joining in between, so you're like missing out whatever things have been discussed previously, right? And that is why I was like keen to get some queries, because many of you have joined late, so you might have like not be able to link what is going on. It's totally fine. I'll share with you the notes and you guys can have a look, and later on if you have queries then let me know. Let me quickly take your attendance. So I would request you to simultaneously go through the notes, don't keep it till the end moment where the final quiz will be there. Just as soon as I share the notes with you, like get some time in your busy schedule, like maybe 1 hour or maybe 1.5 hour, to briefly go through those notes, because the syllabus for the coming quiz is going to be big, and I don't want that you feel burdened by it. Okay, the questions, don't worry, I won't keep it very very hard. The difficulty of the questions will be same as the difficulty you have been seeing in the previous quizzes, but still, like, don't feel too much burdened. So for that you have to like go along, okay, whatever has been taught. Just a suggestion.
Okay, ma'am.
Yes, ma'am.
So who all are present? Murli, is he there? I don't. Murli, are you there? No. Um, Vaishnavi, Shambhavi, Kirtan.
Yes, ma'am.
Okay. Janvi,
Yes, ma'am.
Vani,
Yes, ma'am.
Harshita, Harshita, are you there? Okay. Aditya. Okay.
Yes, ma'am.
Okay. Okay. All right. Maybe Harshita's mic may be not working or something like that. Okay, I'll mark for her anyway. All right, then thanks for joining today. I'll see you in the next class. I'll inform in the group when the next class will be there. All right. So yeah, you guys can leave the meeting.
Thank you, ma'am.
Thank you, ma'am.